I think we'll get started. So today we are going to go over glycolysis or glycolysis, however you want to say it, beta oxidation and the Krebs cycle. So what you need to understand is that the ultimate goal of the cell is for energy metabolism is to produce ATP. So remember that without ATP, the cell can't function because it's ATP that provides the energy for all of the enzymes and the processes that go on. Okay, so so quickly we'll go over glycolysis, just an overview. Glucose is broken down into two pyruvate molecules. And how many ATP do we produce net? Yep, 2. And we also produce 2 NADH, right? So NADH is nicotinamide adenine dinucleotide. Do you guys remember which B vitamin that came from?
Niacin.
Yep, that's right. And what number is that? Yep, that's right. So that's niacin, B3. It's pretty easy because, not easy, but like you can remember it because they both start with the letter N. Okay. And this glycolysis occurs in the cytoplasm of the cell. So you don't need mitochondria and you also don't need oxygen. Red blood cells and eye cells can only use glucose for energy, and that's because they don't have mitochondria. And the other pathways for producing ATP all occur in the mitochondria. And so in order for glycolysis to occur, there must be NAD plus available. So remember, NAD is the electron carrier, and in order for glycolysis to continue, there has to be electron carriers that aren't already full, right? So NAD plus is when it hasn't got electrons, and NADH is when it has electrons. So the first part of glycolysis is the activation steps, and that's activating glucose. So the enzyme is hexokinase. So that phosphorylates glucose and puts a phosphate group on that sugar. And so that performs two functions. So firstly, it's going to prevent the glucose from being able to leave the cell, because remember that these glucose transporters are very specific. Okay. So if you have this, so this is your, if this is your glucose transporter, and here's your glucose molecule, it can bring it in, but once it's got a phosphate group attached to it, it will no longer fit in that glucose transporter. So it can't leave the cell anymore. Okay. So that's an important step. That also activates. It's the first step of activation. So remember that phosphate groups are negatively charged, so they're going to have. They're going to have 2 negative charges. So that's the first step of activation, and that phosphate group is coming from an ATP. Okay, so you have ATP. So adenosine triphosphate. So you can see there's 3 phosphate groups, and it gets convert into ADP, or adenosine diphosphate. Okay, so that phosphate group has been put onto the glucose molecule. Okay, so it's an investment step. You've got to put in ATP to get ATP out. And so in the liver and pancreas cells, instead of hexokinase, you have glucokinase. You don't need to remember too much about it, but just remember that glucokinase has a higher Km. So remember, if you have a higher Km, it means that you have lower binding affinity, right? So that means that this enzyme will only be active at a higher blood sugar level than hexokinase, okay? And that's important for its function, okay? So phosphofructokinase is the second enzyme that's important. That adds another phosphate group to the 6-carbon sugar. So remember that glucose is a 6-carbon molecule, and that's very important to remember. Just remembering how many carbons are in each structure as you go along. And so that's going to put 2 phosphate groups on this glucose molecule, okay? And we'll go through that again in a moment. So that's using up another ATP. So the activation step uses 2 ATP. And then you're going to turn that 6-carbon molecule into 2 3-carbon molecules. And then that 3-carbon molecule is called G3P. And that gets oxidized. And then G3P is oxidized into something called 1,3-BPG. Now, that will be familiar to you guys because remember that BPG can be converted into 2,3-BPG. And what does BPG do? Yep, that's right. So it binds to hemoglobin, and it's going to inhibit or stabilize the T state of hemoglobin, right? So you can see how those two are related. And then the production of ATP, so this 1,3-BPG is unstable, and then it will, it has lots of energy, and you can use that energy to make 2 ATP per 3-carbon molecule, and since you have 2 3-carbon molecules, that's a total of 4, and the end product, we're left with pyruvate, okay? So in the end, ATP, you invested 2, you produce 4, and you have a net gain of 2. You have 2 NAD plus converted to 2 NADH, and 1 glucose converted to 2 pyruvate. So we'll move on to the next page where I've got a diagram. So I just want to go through all these steps again, but using the diagram. So at the top, we have our blood. So in the bloodstream, we're going to have our glucose molecule. Here's glucose. And then here's our cell membrane. And so this is our GLUT transporter. And so glucose is a 6-carbon molecule, 1, 2, 3, 4, 5, 6. So this is glucose. Okay, and then the first thing that happens is it's going to be picked up by hexokinase. So this here is catalyzed by the enzyme hexokinase. And it's going to use up an ATP. So ATP turned into ADP. Okay, so now you've still got a 6-carbon molecule, 1, 2, 3, 4, 5, 6. So it's going to put it on the 6th carbon, on this last one here. Doesn't really matter. Remember that you guys get a diagram in your exam, so it doesn't really matter too much about remembering the names, but you should know what each step is significant for. So this first step is very important. Okay, and now we've got a negative charge on one end of our glucose molecule, and it's called glucose 6-phosphate, or G6P. And then we have a rearrangement. This one's not too important, so I won't spend too much time on it. It just basically gets turned into fructose 6-phosphate, but this is just a rearrangement. It's just allowing the next reaction to occur. So this is now fructose 6-phosphate, and then this next one is important. Okay, so this one's catalyzed by phosphofructokinase. Okay, so that's in the name. It's this is a phosphofructose. It's a fructose molecule with a phosphate attached to it, and it's a kinase, so it'll put a phosphate group on it. And this is also going to use another ATP. So we've got ATP being turned into ADP. Okay. So then we have 1, 2, 3, 4, 5, 6. We have a phosphate molecule on this side, on the 6th carbon, and a phosphate molecule on the 1st carbon. So 2 negative charges on each side, and it's now called fructose 1,6-bis phosphatase. Okay, so remember that. Just a little reminder that if it's bis phosphatase, bis phosphate, if there's an S in there, it means the phosphates are on different carbons. Okay, and if it's a diphosphate, so there's no, or you could say biphosphate, but usually we say bi, so you can say bi or di, but if there's no S, then that means the two phosphates are connected, right? So that's just the difference. Okay. So this is fructose 1,6-bis phosphate, because the phosphates are on the other side. So I'm just going to rub this out. And I'm going to start again from the top. So we've got our 6-carbon molecule, and we have our phosphate group here, which is negatively charged, and our phosphate group here, which is negatively charged. So this molecule here is very high in energy. So why do you think it's unstable? So what you want to do is you want to split this molecule into 2, right? So if there's a negative charge here and a negative charge here, are they going to attract or repel? They're going to repel, right? So if these guys are trying to get away from each other, they're going to put tension on this bond, right? So that makes it really easy for that makes it really easy for aldolase, the enzyme, to come in and just break it. So aldolase is going to come in and it's going to turn it into 2 molecules. Okay. So one molecule will be DHAP, and then that will be converted into glyceraldehyde 3-phosphate. So don't really worry too much about this one. Just remember that this one also turns into glyceraldehyde 3-phosphate. Okay, so this one here is a 3-carbon molecule, and it has a phosphate. So it's G3P, and it's going to be the same on this side. So carbon, 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 phosphate. Okay, so it's a glyceraldehyde with a phosphate on the 3rd carbon. And then, so I'm going to just go down here first. We're going to have the next step. So we're going to oxidize this molecule. So we're going to take the electrons off G3P. So it's NAD+. So NAD+ is always like, oh, I want to take electrons off someone, and then it's going to see glyceraldehyde 3-phosphate, and NAD+ is going to come and take it from G3P. So it's going to become NADH, and at the same time, it's going to a phosphate group. So there's, in the cell, in the cytoplasm, there's phosphate groups. There are phosphate groups just floating around. You've got like these phosphate groups. They're just floating around, and so one of these will be attached. So it's important to remember that this step is using an inorganic phosphate. It's not coming from ATP, so remember that. And then, so then it's going to still be a 3-carbon molecule. This is the original phosphate, and we've put on another phosphate. So now it'll have 2 phosphate molecules, and remember that these are both negatively charged. So it's going to have even more energy than before, because these 2 phosphate molecules are negative and they're going to repel, and they're very close to each other. So this one's called 1,3-BPG. Okay, so what we can do now is we've created this high energy molecule. We can now start to get a return on our investment. So we're going to use this high energy and take an ADP molecule, so adenosine diphosphate, and transfer one of these phosphate groups onto the ATP. So then you are left with this molecule here. Okay, and this is called just phosphoglycerate. Right. And then you can, and then so this one just gets rearranged. Again, this one's not important. Okay, so it's just a rearrangement, and it gets turned into phosphoenol pyruvate, or we can just call it PEP. And then in the last step, we're going to chuck that phosphate molecule onto an ATP. So we'll get an ADP and we'll create an ATP. Okay, so then you're going to end up with a 3-carbon molecule which has no phosphates on it, and that's called pyruvate. Okay, so that's basically glycolysis. Now, at this stage here, 1,3-BPG, this can go down an alternate pathway. So as part of the regulation of glycolysis, it can be, so this is a reversible reaction. It can be turned into, so the phosphate group can be moved from the first carbon to the second carbon. So that's 2,3-BPG, BPG, 2,3-BPG. And this is the one that's going to bind to hemoglobin in the center of the tetromere. Remember? Okay. Okay, so now I just want to talk about what can happen if you have arsenate in the cell. So if you have arsenic poisoning, you're going to have arsenic or arsenate going around in your bloodstream. And if you look at the diagram on the right side of your piece of paper, you can see that it looks very similar to phosphate. So here's our arsenate molecule. And it's going to be negatively charged, just like phosphate. And so when we have this reaction occurring where G3P is oxidized, NAD plus is going to come in. Sorry, this should be to the right. So in the diagram, I've got it going to the right. So you've got NAD plus is still going to come in, and it's going to take the electrons and going to become NADH. But this time, instead of using a phosphate, it's going to use this arsenate, and it's going to be put on the on this G3P. So you're going to end up with this molecule here, which is which has got arsenate on it instead of phosphate. Okay, so this one's called 3-phosphoglycerate. You don't have to remember that name, but just realize that there's an arsenate, and since these are both negatively charged, both of these are negatively charged, it's unstable. Okay, so it wants to get rid of that arsenate, and that arsenate is just going to come off. So the arsenate's going to come off the molecule. But remember that, so it's gone from G3P to this molecule, and then it's gone to, so it's going to become phosphoglycerate now. So we're at this step. But remember that in this step here, we created ATP, but we've skipped this step when we created this arsenophosphoglycerate. Okay, so then if you go down this pathway, you're only going to make one ATP per, so you'll go down this pathway and you'll make one ATP because you've missed one of your chances to make ATP. So then if that happens for both sides, then you put in 2 ATP and you're only getting 2 ATP out, so there's no net gain of ATP. So the cell, remember, it's trying to do glycolysis in order to make ATP. So if you're a cell that relies solely on glycolysis for energy production, then you're going to run out of energy pretty quickly. Okay. So then let's just talk about the fate of pyruvate. So at the bottom on the left side, let's have the aerobic pathway. So you have pyruvate. So let's say on the left side it's with oxygen. So pyruvate will... So if you have oxygen, what happens is that this NADH, the NADH here, so you have the mitochondria, and the NADH will go to the mitochondria through the electron transport chain, and then it will be converted back to NAD+. So if you have oxygen and you have mitochondria, you're going to get NAD+ being regenerated, okay? And remember that we need to have this NAD+ in order for glycolysis to occur, okay? And if you have oxygen, that's happening, so we don't have to worry about regenerating NAD+, sorry. So in this case, if you have oxygen, pyruvate is going to be oxidized again. So you're going to get NAD+ turning into NADH. And you're going to have a coenzyme A molecule. So coenzyme A comes in, and this is pyruvate is a 3-carbon molecule. And you're going to get rid of one of these carbons. So you're going to have a carbon dioxide molecule coming out. And you're going to have a 2-carbon molecule. So this part is called acetyl. So this is an acetyl group. So that's 2 carbons. And that's connected to our coenzyme A molecule. So this is acetyl-CoA. All right, and so if you remember from last week, that S is the sulfur group, and the sulfur group is the carrier, is where it binds, right? So this is acetyl-CoA, that's if you have oxygen. And then the acetyl-CoA can go into the mitochondria, but this occurs down in the cytoplasm. And then on the right side, let's draw what happens if you don't have oxygen. So you have pyruvate, which is a 3-carbon molecule. And if you don't have oxygen, so without oxygen, then this NADH, the NADH can't go to the mitochondria. This step can't occur, so you have a whole heap of NADH which is building up, so you need to get rid of this NADH and regenerate NAD+. So we're going to put the electrons from NADH back onto pyruvate. So you've got NADH putting the electrons back on to pyruvate. So now this is still a 3-carbon molecule, and this molecule here is now called lactate or lactic acid. Okay. Cool. Yeah, so it's a bit long, but glycolysis is like a real important one to understand. And if you can understand that, that makes the rest of the course easier. Okay, so we'll move on to beta oxidation. So we have, inside our adipocyte, we have hormone-sensitive lipase. And this enzyme breaks down stored triglycerides into fatty acids and glycerol. So remember, adipose tissue is just fat. The fatty acids travel in the bloodstream and they're bound to albumin. But the glycerol is polar. So it can travel on its own in the blood. And the fatty acids can be picked up by various tissues, including the heart, muscle, and liver. So we have the adipocyte. We have the triglyceride. And this reaction here is catalyzed by hormone-sensitive lipase, or HSL. And it's broken down into glycerol and 3 fatty acids. Okay. And we'll go through this more later, but you have insulin. So if you have insulin, when is insulin released? When your blood sugar is high or low? Yep. So if you have lots of sugar in your blood, you'll get insulin released. So when you have insulin, that means you have a lot of sugar. And if you have a lot of sugar in your blood, do you need to have fatty acids? No. So insulin will tell hormone-sensitive lipase, we have a lot of glucose available, so we don't need you to be working so hard. So you can just quiet down, and it's going to inhibit hormone-sensitive lipase. Okay. But on the other hand, we've got adrenaline, and we've also got glucagon. So glucagon is released when blood sugar is low. So if there's not a lot of sugar around, we need to have an alternative source of energy. So then glucagon will tell hormone-sensitive lipase, we haven't got a lot of glucose, so can you start working a bit harder, and so that we can use the fatty acids for energy. And so it's going to stimulate hormone-sensitive lipase, and also adrenaline. So adrenaline's released when you have that fight or flight response. So you want to mobilize all your energy stores so that if you have to run away, you have a lot of energy available. So adrenaline is also going to stimulate hormone-sensitive lipase, and we'll go through that a bit more towards the end of the BIOC in the next few lectures. So then you have the bloodstream and glycerols and fatty acids going to diffuse into the bloodstream. So glycerol is polar, so it can just travel on its own, but the fatty acid is non-polar, so it's going to have to be bound to a large protein molecule, which acts as a carrier in the bloodstream. So albumin is like a big protein. It's a very big protein, and it just acts like a bus for fatty acids. So fatty acids will catch a ride with albumin and then get to wherever they need to go and then jump off. So I think we'll go to the next page first, and then we'll come back and finish off that page because it'll be easier to fill it out once we've gone through the diagram. So at the top, that's basically just the same thing. You had your adipocyte, you had your triglycerols being turned into glycerol and fatty acids, and that's being catalyzed by hormone-sensitive lipase, right? And then they're going to diffuse into the bloodstream. Glycerol and fatty acids. So the glycerol will usually just go straight to the liver, okay? And then the liver can turn it back into glucose. Okay, so you have three. Remember, glycerol is our 3-carbon molecule. So if you put two 3-carbon molecules together, you can make a 6-carbon molecule, which is glucose, right? So glycerol can be turned into glucose. And we'll go through that more later on as well. And you have these fatty acids, which are going to be bound to albumin. And they're going to travel in the bloodstream. And then you have this big, in this diagram I've made it big just so that we have enough space. So this is the cell. So this is our cell. And the fatty acid is going to be taken up by the cell. And so now once this fatty acid is inside the cell, it has to be activated. So we have our fatty acid. And it's going to be, it has to be put on to a coenzyme A molecule. So let's see. So we have, so this fatty acid is going to be combined with a coenzyme A molecule, okay? And sorry, once it's attached, once the fatty acid is attached to anything, it's now called an acyl group. So if it's on its own, it's a fatty acid. If the fatty acid is attached to something else, it's now called an acyl group. Okay, so this is acyl-CoA. And in order to do this, we require ATP. So this, whenever you put a coenzyme A on something, it requires energy. So we're going to take ATP, and usually ATP gets turned into ADP, or adenosine diphosphate, but in this case, it's going to get turned into AMP, adenosine monophosphate. Okay. So that means it's going to lose 2 phosphates. So it's the energy equivalent to using 2 ATPs. Okay. So we've got our acyl-CoA, but then there's this really complicated step afterwards where we have a problem where the mitochondria has 2 membranes, right? So there's an outer membrane and an inner membrane. And so the outer membrane is quite permeable and it can let quite a lot of stuff through, but the inner membrane is very, very selective and it's not going to let as much stuff through. So this is our So this is our outer membrane of the mitochondria. And so it's permeable. So it has a protein. It has a transporter on the outer membrane, which will let the acyl-CoA come in through the outer membrane. So we have our acyl-CoA, which has come in. And we also have our, so this is our, so this is our inner membrane. Okay. So the space in between the outer and inner membrane is called the intermembrane space. I'm sure you guys know that, but just making sure. So this acyl-CoA can't get through the inner membrane, so it has to be moved to a carnitine. So there's a carnitine molecule, and this is catalyzed by a protein on the inner surface of the outer membrane. So it's on the inner surface of the outer membrane, and that's called carnitine acyl transferase. So it's going to transfer the acyl onto the carnitine, so it's called carnitine acyl transferase 1, or CAT1. So it's going to be turned into, so now it's going to be turned into acyl carnitine, and you have your free coenzyme A molecule. Now there's a transporter on the inner membrane which can allow acyl carnitine into the cell. And so once it's inside the cell, once it's inside the inner membrane, so you've got acyl carnitine, it's going to get put back onto the coenzyme A. So the one that transfers the acyl back onto the coenzyme A is called CAT2, or carnitine acyl transferase 2. And this is on the inner membrane of the, the inner surface of the inner membrane. I don't know why they made it so complicated. So now you have acyl-CoA again, and your free carnitine. Okay. So there's a lot of it is a lot of detail, but they do expect you to know it. Okay. So just make sure that you understand that. Okay. So now we'll go through beta oxidation once the acyl-CoA is inside the cell. So I'm just going to make this a bit bigger. And all of this is going to be happening inside the mitochondrial matrix. So we have our cell, we have the outer membrane of the mitochondria, and we have the inner membrane of the mitochondria. So we are inside the mitochondrial matrix, okay? So you're going to have to write this quite small, because I'm going to go through, we're going to use a 6-carbon fatty acid to just demonstrate what goes on. So we have, we're going to draw a 6-carbon fatty acid. So 6 carbons, and we'll have our CoA attached to it. So it's an acyl-CoA. So this last carbon here will have a double bonded oxygen. Oops. So double bonded oxygen. And then the second carbon is our alpha carbon. Okay. So alpha is like the first carbon. We don't count this carbon. And so then this one here, the third one, will be the beta carbon. Okay. Okay. So I think now you can see why it's called beta oxidation. So this is our acyl-CoA. And then we are going to oxidize it. So up till now we used NAD plus to oxidize, but this time we're going to use FAD. So we have FAD, which is, it doesn't want electrons as much as, it's not as good at taking electrons as much as NAD plus, but it'll still do so. So FAD will be turned into FADH2. Okay, so there's a difference there. NAD plus goes to NADH, but FAD goes to FADH2. So again, we're going to draw our 6-carbon molecule, 3, 4, 5, 6, and it's attached to CoA. And this first carbon here has a double bonded O. This is our alpha carbon, our beta carbon. And so between the alpha and the beta carbon, so we've taken electrons away from this carbon here, the beta carbon. So if you take electrons away from it, now it has to share with someone else. So if it has to share with someone else, it's going to create a double bond between the alpha and the beta carbon. Okay, because remember that each carbon wants 8 electrons around it. If you remember from Chem 191. So it's now asked the alpha carbon, can I have some electrons and share with you? So we have a double bond. Now we are going to add a water molecule. So we're going to get H2O, and this is called hydration because we're adding a water molecule. So again, we'll draw our 6-carbon molecule, 1, 2, 3, 4, 5, 6, and we have our CoA. So this one is a double bonded O. This one was our alpha carbon, and this one was our beta carbon. So we now have a hydroxyl group, an OH, added to the beta carbon. Okay, so this, and the double bond is gone now. So we've added the water, and we've got an OH group on the beta carbon. And then we're going to oxidize it again, and this time we're going to use NAD+. So NAD+ is going to come in and take the electrons, and it's going to take it from the oxygen this time. So then you have, oops, I'll draw it a bit lower, I'm going to run out of space. 2, 3, 4, 5, 6, and then the CoA. Okay, so we have our double bonded oxygen on that first carbon, the alpha carbon, the beta carbon. So remember the beta carbon had the OH group. Now it's going to have a ketone group, or the double bonded oxygen. So that's a ketone. So then we can now cut here. And if we do that, we are going to end up with 2 molecules. So the first one is going to be, I'm just going to switch the positions of these two around just so for the next step. So we're going to have this part here, which is 2 carbons long. So now this is, so we've got an acetyl-CoA that's been created. And we've also got a 4-carbon molecule. So we've got a 4-carbon acetyl-CoA, which is now, which is 2 carbons shorter. Okay. So now this one's going to undergo another round of beta oxidation. So we've got us, now it's our 4-carbon molecule, CoA. So remember, our first carbon has a double bonded O, and the second one is our alpha carbon, and the third one is our beta carbon. It's going to get oxidized. It's going to get oxidized by FAD into FADH2, and that's going to create, so we have our 4-carbon acetyl-CoA. This is our double bonded oxygen, alpha, beta. So we're going to create a double bond between the alpha and the beta carbon. Right. And then we're going to add a water molecule. So this one has the oxygen, this is the alpha carbon, beta carbon, and it's going to add the OH onto the beta carbon. Right. And then it's going to get oxidized again. So this time it's going to get oxidized by NAD+. So 4 carbons and a coenzyme A. We've got a double bonded oxygen on the first carbon, a double bonded oxygen on the beta carbon. Okay, and now it's ready to be split again. So we're going to split it here. And we're going to have so when we split it, we have to have another coenzyme A coming in as well. I forgot to mention that for this one here. So we'll have another coenzyme A that's floating around come in and join. So then we'll have we're going to have another. So in the last round of beta oxidation, you're going to produce 2 acetyl-CoAs. Okay, so for this, says everyone, I'll just rub out this side. So for our 6-carbon fatty acid, how many rounds of beta oxidation did we do? Yep, 2. So 6 carbons, fatty acid. We performed 2 rounds of beta oxidation. Okay, so if you want to know, and they're going to ask you this in the exam, so they're going to ask you if you have a 10-carbon fatty acid, how many rounds of beta oxidation will you do? So it's divide the number by 2 and then minus 1. Okay. So in this case, 6 divided by 2 is 3, minus 1 is 2. So you're going to do 2 rounds of beta oxidation. And if you turn 6 carbon into 2 carbon units. That's going to give you 3 acetyl-CoAs. And remember that each time you do a round of beta oxidation, you gain 1 FADH2 and 1 NADH. So you're going to have 2 NADH and 2 FADH2. Okay, so for every round of the number of NADH and the number of FADH2 that you produce will be the same as the rounds of beta oxidation that you do. So this part is important. Okay, they're like definitely guaranteed to ask you that in the exam. They pretty much do every year. So make sure you understand how many products will be made from beta oxidation. So then I was supposed to go through citric acid cycle as well, but we're like pretty much out of time. I don't mind staying for another 10 minutes to go through the citric acid cycle. Do you guys mind? Should I keep going? Okay. So we have the citric acid cycle. The citric acid cycle is also known as the. So this is oops. So this is citric acid. You don't need to remember this, but just for interest, citric acid is also called tricarboxylic acid. So it means it has 3 carboxyl groups. So remember, a carboxyl group is this one. So there's 3 carbons with 3 carboxyl groups attached. So that's citric acid. And there's also a hydroxyl group here. But that's just why it's called tricarboxylic acid. That's the name. And so it can also be called a tricarboxylic acid cycle. But that's not too important. I just wanted to show that because some people got confused why it's called the tricarboxylic acid cycle, or TCA. So first of all, we're going to start off with our acetyl-CoA. So this acetyl-CoA could have been produced from glycolysis. So it could have come from pyruvate, or it could have come from beta oxidation. Okay, so you've got pyruvate or your fatty acid. Both of them can be turned into acetyl-CoA. Now, remember that this is a 2-carbon molecule, and we have something called oxaloacetate, which is a 4-carbon molecule. And it's going to combine. Acetyl-CoA is going to combine with acetyl-CoA. So you're going to put 2 carbons and 4 carbons together to create a 6-carbon molecule. And this is citrate. 5, 6. So this is citrate. And then it's going to be rearranged. And this one is important to remember. So the enzyme that catalyzes this step is called aconitase. So it's important to remember this one, and I'll tell you why. Oh, well, you can see why on that diagram. So it gets rearranged, but it's still 6 carbons. And this is called isocitrate. So it's just an isomer of citrate. And then our next step, we are going to oxidize isocitrate. So we're going to have NAD plus coming in, and it's going to take electrons off, and it's going to take electrons off isocitrate. And at the same time, it's going to take one of these carbons off. So you're going to have a carbon dioxide coming off. So now we've gone from 6 carbons down to 5 carbons. So this is called alpha ketoglutarate. And then the next step is pretty much exactly the same. So again, it's going to get oxidized by NAD+. So NAD+ is going to take electrons and turn it into NADH. And it's also going to get rid of one of the carbons, so we're going to lose a carbon dioxide. And since we've lost a carbon, it's now going to be a 4-carbon molecule. Okay, but there's one more thing. So in this step, we have a coenzyme A coming in. So we have a coenzyme A that's joined on to our molecule. So now it's going to be joined to a coenzyme A molecule. So this is called succinyl CoA. All right. So remember that if you put a coenzyme A on a molecule, it makes it higher in energy. So this one's, if it's joined to, so that's the whole reason why we put coenzyme A onto these molecules. It makes it higher in energy. Okay. And so we can use this energy to phosphorylate so remember that in the cytoplasm we had phosphate groups just floating around. It's the same in the mitochondria. We have phosphate groups floating around as well. So we can take a phosphate group and utilize it in this reaction. And we're going to take a GDP, so guanosine diphosphate. It's pretty similar to ADP, and it's going to put that phosphate group onto the GDP and turn it into GTP. Okay, and this GTP is pretty much equal to ATP, so you can just think of it like ATP. And we're also going to lose that coenzyme A, and it's still the same molecule, but it's lost the coenzyme A, so now it's just called succinate. And then the next 3 reactions are very similar to what we just went through before. So the 3 steps of beta oxidation, where we had oxidation by FAD, and then the hydration, and then the oxidation by NAD+. So that's exactly what happens here as well. So we're going to have FAD, oxidizing succinate, And that's going to turn succinate into fumarate. Sorry. Yeah, fumarate. So there'll be a double bond here. And then we'll have a hydration. So we'll have water coming in. Oh, sorry, I forgot to mention. So this step here, succinate to fumarate, is a little bit different. So this one happens on the inner membrane of the mitochondria. So all of these other reactions occur in the mitochondrial matrix, whereas this one is on the inner membrane of the mitochondria, on it's complex 2 of the electron transport chain, and we'll go through that next week. And so once we've added the this is now malate. And then we're going to oxidize it again, so NAD plus to NADH. Okay, so remember that this is very similar to the 3 steps of beta oxidation. It's pretty much exactly the same. Oops, sorry. This should be, going all the way here. Okay? So that's the citric acid cycle. And if you want to remember all the substrates in the citric acid cycle, you've got a little mnemonic at the bottom there. So that citrate is Krebs starting substrate for making oxaloacetate. Okay, just a handy mnemonic if you ever want to go to a party and be pretty cool. So I'll just post up the answers, but try and fill it out if you can. So fill out all the gaps, but I'll post the answers later on Facebook. Okay, sorry I went a little bit over time, but hopefully that helped. All right, thank you.
After that,